Research Kaleidoscope. And first up, our first speaker tonight is going to be Michael Chen, and he's going to speak on introducing URK and solving the structure of DNA triplet repeats that cause disorders such as Friedreich's ataxia.
That's great.
And Huntington's disease. All right, so Michael, you're up.
Hey, I'm Michael Chen, and welcome to the Undergraduate Research Kaleidoscope. This is the first time this event has been sponsored by the Tower, URK, and the library. And so the Undergraduate Research Kaleidoscope represents something that's been in the works for quite a while. And it's usually every semester, but last semester it sort of died out. And the reason we want to sponsor this, well, first let me talk to you about the Tower Undergraduate Research Hall. The Tower is a student-run publication whose purpose is to showcase the amazingly varied and impressive undergraduate research work here at Tech. And so our first attempt at doing this was with the inaugural issue. And the groundwork was laid for this issue by a group of undergrads who firmly believe that without communication, research is meaningless. This is our second issue, which is due to come out any day now, probably tomorrow, we'll see. But this is a continuation of the original goals of the Tower staff. And we're looking to continue publishing every single semester with print and online editions. So the purpose of the Tower is to bridge the two worlds of technical research and mass communication. But we're also looking for more ways to do that other than our online and our print edition. And so it is our hope that one day we'll be able to communicate research as easily as we chat with our friends and as casually as we talk with one another. And I believe, and the staff at the Tower believe, that the 20 by 20 format of the Pecha Kucha is a great way to increase accessibility to research, because research is a very vital endeavor for every society on Earth. And so I'd like to kick off the undergraduate research kaleidoscope by starting off with research that I do in Professor Nicholas Hugg's laboratory. Friedreich's ataxia is a hereditary neuromuscular degenerative disease that results in loss of heart and muscle function in the individual suffering with this. And so researchers have noticed that Friedreich's ataxia is caused by a lack of mRNA transcripts in certain genes, vital genes. But they don't know why this is. And so they did a sequence analysis of the genomes of these individuals, and they found that these individuals had huge expansions in DNA triplet repeats, meaning that 3 bases are repeated over and over and over again, hundreds and hundreds of times in the human genome. And researchers also know that these triplet repeats can form several different types of unusual DNA structures, including on top of the double helix, these can also form DNA triplexes. And researchers think that these unusual DNA structures are what cause the transcription inhibition. And so what we're asked, the question we want to ask is, what structure is in the cell that causes a decrease in mRNA transcripts? And so the way we do this is we use a well-known mode of binding to DNA called DNA interpolation. Now this is where a small planar molecule binds between the base pairs of nucleic acid. And this is a very specific interaction. Only certain interpolators can bind to certain types of nucleic acid structures. So what we did was we genetically engineered yeast to express these long DNA triplet repeats that you see in individuals with Friedreich's ataxia. And then we introduced intercalators to the yeast system to see if we could induce chromosomal breaks. Now, chromosomal breaks occur when a intercalator binds at a certain spot, and that causes that spot to become more fragile. And when that spot becomes more fragile, they're likely to break more often. And so, we found that when we introduced intercalators, chromosomal breaks occurred only for the ASA3 and ASA5 intercalators. And so, we don't know what structures these intercalators target. All we know is that these intercalators bind to some sort of DNA structure within the cell, and then they cause chromosomal breakages. So we need to deduce the structure. We do this by using a method called equilibrium dialysis, where we put a nucleic acid of a known structure inside a membrane, a semi-permeable membrane, and then we put all of this into a solution with intercalators. And we can measure the amount of intercalator that goes through that semi-permeable membrane. And once we know that, we know the affinity of that intercalator for a certain type of nucleic acid structure. And so we discovered that it was a DNA triplex that ASA3 and ASA5 bind to, and that this DNA triplex causes Friedreich's ataxia. And since we've discovered that structure, we hope that we can hand that over to translational researchers who can then tailor medicines to treat Friedreich's ataxia. We also think that we've discovered a novel method of investigating DNA disorders within human beings by using the specificity of these intercalators. We can better understand the different types of nucleic acid structures operating within our systems and possibly create new medicines to treat more diseases. So I hope you guys enjoy the undergraduate research kaleidoscope and enjoy the rest of the presentations.